Welcome to the Biomedical Engineering Department here at the New Jersey Institute of Technology. A good number of our biomaterials laboratories are right here at the Life Sciences and Engineering Center. So with that, why don't you come on with and let's take a look at some of those laboratories. Here at the Life Sciences and Engineering building, we have a variety of laboratory spaces that offer highly intercollaborative work, like the one you see behind me. In addition to a variety of wet lab spaces, we have dry lab spaces right above, which offer, again, highly collaborative work. Additionally, here at the Science Commons, we have a wide variety of spaces and areas, including presentation screens for students to come in and learn. So with that, why don't we go up to the third floor and take a look at my laboratory. Here at the Life Sciences and Engineering Center, we have a number of collaborative spaces, as you can see here and behind me, where students can come do homework, relax, or even faculty can meet and write some grants. Additionally, on the second floor, like the first floor, we have highly intercollaborative wet lab spaces, as well as some of the laboratories from the chemical engineering department. So let's go up another floor. We're standing on the third floor of the Life Sciences and Engineering building. And we're right outside my laboratory, and as you can see here, we have a number of students in my laboratory who do amazing research. Ranging from undergraduates, graduate students, as well as postdoctoral students, we work on self-assembling peptides. So these short peptides, which I'll tell you all about as we enter into my lab. So coming into here, we have my postdoc, Peter Wen, who kindly opened the door for us. And the lifeblood of our research here in the KumarLab is really the solid phase peptide synthesizer. Really, you know, if we had the lights off, we'd be able to see very clearly how each amino acid, each short building block of a protein comes together step by step like Lego to form a polypeptide or a short protein. We're able to make these peptides. Now let's go take a look at how we process them. Continuing through the lab, we have a variety of different characterization tools as well as a number of different tools that we can use to program syntheses, characterize peptides, measure fluorescence, luminescence, bioluminescence. And of course, as engineers, we love to play around with some electronics. So here we have Peter working with an Arduino board as well as a pump to essentially create an artificial flow loop mimicking the circulatory system in the body. Next, let me show you some of these hydrogels. So we can take these peptides that are made in a variety of different ways with a variety of different sequences, dissolve them in aqueous solvents, just like water or salt solution. And what we see is that they form hydrogels, just like this. What's unique about these hydrogels is that they are force responsive, kind of like toothpaste, kind of like hand sanitizer. When you put a force on it, it flows, and when you relieve that force, it stays in place. So you see this hydrogel stays in place. When you put a force on it, it flows just like that, and then you can tap it to bring it all together, and almost instantaneously, it forms that hydrogel again, trapping those bubbles. Next, what we're going to do is take a look at how these materials, these hydrogels, interact with cells. What we see here are dental pulp stem cells. These stem cells, or progenitor cells, have the ability to form a variety of different tissue. What we're interested in is to take our hydrogel materials with specific signals and see whether we can tailor the phenotype or determine what these cells ultimately become. What we see here is a stuffed mouse and a stuffed rat. And what's interesting is that we take these hydrogel materials and we inject them into the backs of these animals. Our animal facilities are just down the road at Rutgers Newark. We make a little tent in the back of a rat, inject it subcutaneous or underneath the skin. Now, I told you about a dental peptide, which is what I have in my hand right here. But what we have in our laboratory are a variety of different students working on dental applications, neurogenic, cell delivery, vascular grafts, and a variety of different applications. If you're interested in medicine, in biomaterials, or biomedical engineering, or any combination of those, please feel free to come reach out to me. My email address is vak@njit.edu. My office is always open.